Hello, everybody, and welcome to the latest installment of the CDX Engage webinars hosted by Covance by LabCorp. This webinar is entitled Tumor-Specific 3D Cell Culture Models for Accurate Testing of Anticancer Agents. This is an online event designed to bring you with the opportunity to explore the advantages of a 3D culture approach over traditional methods. To briefly introduce myself, I am Noemi, a member of the CDX Engage team, and I will be chairing today's session where we will be joined by two expert speakers from Covance by LabCorp and cPredicta. The webinar today will consist of two presentations, followed by a Q&A where you will be able to ask your pressing questions to the speakers. Our expert speakers joining us today are Julia Kirchner, founder and CEO of cPredicta, and Sumitra Urs, scientist of scientific development at Covance by LabCorp. I'm sure you will all agree that our speakers today have wealth of knowledge in the field. Please submit all questions via the Questions tab at the bottom of your webinar dashboard. You can submit this at any time throughout the webinar. We will try to answer all questions in the allotted time, but all unaddressed questions will be passed along to Covance by LabCorp so they can follow up with answers directly. We will now be moving on to our first presentation by Julia Kirchner, founder and CEO of cPredicta. Julia has over 20 years of experience in oncology research and development, spanning multiple areas of cancer biology, including the development of organotypic preclinical models, molecular target and biomarker discovery, evaluation of drug resistance, tumor microenvironment, cancer stem cells, and tumor immunology. Julia founded cPredicta with the goal to develop tumor-specific 3D culture models, reconstructing the native microenvironment of human tissues to mimic the drug behavior as seen in patients. From target discovery and lead optimization to preclinical evaluation of efficacy and toxicity, cPredicta's goal is to develop the tools necessary to accurately identify compounds that will have the highest probability of improving human health. So now, without further ado, I will share Julia's presentation.
Hello. Today we're going to talk about tumor-specific 3D culture models for accurate testing of anti-cancer agents. I will start off by describing various 3D culture systems and talk about what cPredicta has done to aid in the development of these models for drug development. And then Dr. Urs is going to discuss their data at Covance from the cPredicta's 3D culture systems, and also we'll talk about how these platforms can be incorporated into the drug development platform. So really the reason for developing these new systems comes from the current problem in drug development, and that is that only 1 in 20 drugs that enters the clinic ends up approved and actually sees patients. And this becomes a really expensive and very long-term endeavor to develop even a single drug, because as you can see here, there is a very poor efficacy seen in the clinic from any one of those compounds. So you need at least 20 drugs to actually get one that works. And a number of people have been trying to solve this problem by creating more relevant systems to test the drugs in the lab before they actually get into the clinic. So here I'm going to touch on some of those systems in a brief summary. So one of the systems is a spheroid model where the cells are aggregated within a hanging drop. This can mimic a tumor mass, and cells can grow in this three-dimensional tumor cluster. Various scaffolds have been developed and used, such as matrigel, collagens, hyaluronic acid, they're inert hydrogels, and this helps to incorporate the extracellular matrix into the 3D system and provides this ECM support for cells to form tumor-like structures. Tumor explants provide the next level of complexity in these systems because these really bring together all the cellular components of a tumor and some of the extracellular environment as well, and this allows the cultures to really maintain the setup, so to speak, as what was in the patient. And then with bioprinting, this technology allows layering of multicellular assemblies and creation of multicellular structures that also resemble what happens in a tissue. And finally, organ-on-a-chip technologies introduce microfluidics to allow perfusion of media or other cytokines or any kind of drugs within the cell culture and allowing the cells to form 3D structures and retain the perfusion that is present in an organism. There are, of course, problems with each of these systems as well as benefits. All of them provide a platform that allows cells to form three-dimensional structures, therefore being much closer in architecture and morphology and behavior to the actual tumor compared to the standard 2D culture systems that are available currently and are used widely for drug development. Unfortunately, many of these systems lack human environment. So for example, the hanging drop cultures lack any kind of microenvironment. They're growing in a liquid droplet. Bioprinting usually incorporates hydrogels and other inert extracellular matrix molecules that don't really mimic human tissue. And in general, none of the technologies that are listed here provide true human microenvironment, and lack of this microenvironment affects how the drugs behave in these systems, and they skew, of course, the results of the drug testing because these are not truly physiological setup of the tissue where the extracellular matrix and soluble components match the tissue and the disease of interest. However, for a lot of these systems, such technology can be incorporated, and this is what zPredicta has been focused on, is developing really tumor-specific models and tumor-specific extracellular matrices that can be incorporated widely into a lot of these technologies. And really the premise behind what we've been doing is the fact that each tissue has different type of extracellular matrix, and that really drives behavior of cells. And here what you're seeing is the cryo-EM images showing lung, bone marrow, and liver extracellular matrix. And as depicted here, it's very clear that matrix of the liver and matrix of the lung or the bone marrow really has very different architecture, and the cells will behave themselves very differently. So this is what we took as a guiding post for development of our models, where we would take an organ of interest and really determine what kind of extracellular matrix and what kind of soluble factors are present in there in a disease and healthy conditions, and this is what we really reconstruct. So we aim to incorporate all the elements of tissue microenvironment, as I said, extracellular matrix, soluble factors, and such, and this really reproduces the conditions of environment-mediated drug resistance, and this is what drives a lot of resistance to compounds in patients, so we're able to capture this element of tissue microenvironment. And so we can actually test drugs in a much more accurate system. And here on the right, we're looking at the reconstruction of the bone marrow, our bone system, where the honeycomb structure is the bone marrow matrix, and then you can see a couple of cell clusters. And on the bottom here is the actual ex vivo bone marrow, same thing, honeycomb structure of the ECM, and it's a couple of cell clusters, and these are really virtually indistinguishable from one another. There are a number of different attributes of our system that become really important for decisions of how to set up and which model to choose. So as I mentioned, the technology that we're developing is really predictive and can give an idea of what's going to happen with the drug in a patient. And I'll show some data on that in a second. But we do see high correlation between the clinical response and what we see in culture, and therefore we have high confidence that these models can actually replicate tissue behavior and drug behavior. As I said, these are tumor-specific systems. The extracellular matrix is formulated from natural ECM components, so things like collagens, laminins, elastin, fibronectin. These are the raw materials that we use, and these are mixed in specific formulations to mimic what happens in a tissue. And then disease-specific medium supplements are added that match both the tissue and the condition of interest. We can use various cell types in the system, such as epithelial cells, primary cancer-associated fibroblasts, other stroma, fibroblasts, mesenchymal stem cells, immune cells, as well as primary bone marrow cells. And the format that the cultures take is usually spheroid or tumoroid. Sometimes these tumoroid structures form stellate structures or networks. That really depends on cell identity. And this can be co-cultured with mesenchymal stem cells, immune cells, other stromal cells, or in case of growing primary cells, usually all these stromal cells actually come with the tumor tissue that can then be incorporated. So far, we have worked in both human and mouse, so those systems are available. The platform is optimized for 96-well format. We've used it also in 24 and 48-well format, and it is HTS compatible to 584 wells as well. The setup is the cells are embedded in the ECM, so the cells are actually not growing again on the surface. They're not forced to artificially flatten. They're actually allowed to form whatever physiological structures they normally do, and the average thickness of the culture is about 800 microns, and this can be modulated as needed. We tested a number of different drug classes as single agents or in combination. So we've worked with small molecules, worked with biologics, with antibodies, antibody drug conjugates, trispecifics, or various cellular therapies such as CAR-Ts. And we've looked at multiple readouts such as cell-based assays like cell titer glow, ELISAs, live dead staining, things like that. We can image in situ, so bright field fluorescence, confocal immunohistochemistry can be done, flow cytometry can be done, any kind of omics, genomics, proteomics, and cells can actually be also used for in vivo studies after the culture. Assay time really varies, and it's dependent on the molecule that's being tested. The average culture time that we've been using is about 14 days, although especially with primary cells, it can go as far as 35 days. In some cases, we've done longer, but kind of the maximum's been really around 35 days. And the endpoints are various, again, dependent on the interest in the experiment. Morphology, looking at just spheroid formation, tumor stromal interactions, cell viability, looking at tumor versus non-tumor viability, cell proliferation, or any kind of other readouts are possible. So in terms of the predictive capacity that I was talking about, this is how the study was done. We're looking at multiple myeloma in this case, where in the clinic, patient aspirate, bone marrow aspirate was taken and sent to us. Part of it went for clinical evaluation as well. Once the cells were analyzed in the clinic and the treatment regimen was determined, we treated the cultures that were set up with these cells with the same drugs that the patient would receive in the clinic. And then at the end of the culture and in the end of the treatment period, we would compare the clinical response to response in the culture. We're looking at, does the patient respond? Is the patient resistant? Are the cells sensitive in culture? Are they resistant? Do we see cell kill? And this is the summary of the results that we got. The bottom line is that our system was able to predict 19 out of 21 outcomes correctly. We predicted 8 true positives, so where the cells were sensitive in the clinic and they were sensitive in our system. We had 11 true negative, resistant in the clinic and resistant in the system, no false negatives, and we did have 2 false positives. And on the right-hand side, we're looking at the actual data, as a snapshot of the study, because this is a big experiment with a wide range of treatments, number of doses, and a combination treatment in this case. But we're just looking at a snapshot, and here a patient who was resistant in red. You can see there was really no cell kill in culture, but the patient who was sensitive in the clinic in cell culture, we did see reduced survival of cancer cells. The culture setup is similar to standard cultures with a couple of extra steps added. What we do is we coat a plate with stromal ECM, because most tissues have distinct niche for stroma versus tumor. So we want to capture both of those. So we coat the plate with the stromal ECM. Sometimes it's a thin layer, sometimes it's a thicker layer, really depends on the system. Then the cells are mixed with the tissue-specific ECM, which is overlaid on top of the stromal ECM. The whole thing is allowed to polymerize. And then once polymerized, we add media on top, and media is supplemented, as I was talking about before, with disease-specific supplements. Then we allow the cultures to grow for about 3 to 5 days to just let the cells aggregate or migrate or just establish the structures that they normally do, be it spheroids, be it stellids, be it networks, but just let them behave normally. And then we add therapeutic agents, and really the treatment is dependent, duration of the treatment is dependent on the type of the agent. And then as the endpoints, as I said, we can do imaging, or we can isolate cells from the ECM without use of enzymatic digestion, and then it can go for flow cytometry or omics or in vivo studies or anything like that. So now I'm going to show some examples of our single compartment models. We'll actually have also multi-compartment models, but I won't have time to discuss those, but I'm happy to answer questions about them. And I'll touch on our reconstructed bone and our reconstructed lung system. So as you know, in bone marrow, there's a large complement of various cells. There's hematopoietic cells, there's stromal cells such as osteoblast, osteoclast, making and degrading bone, there's adipocytes. And this really multicellularity in many cases is what drives disease. Multiple myeloma, AML, a lot of these cells are supportive, so you need the whole complement of the cells in culture. So when we take the aspirate from primary bone marrow cells from myeloma patients and put them, say, on plastic in Matrigel or in collagen 1, as shown up here in the top images, these cells really don't survive past a few days. So by day 14, there's really not much left in the system. But when the same cells are cultured in our bone, you can see by day 5, there's significant proliferation of these cells. They start migrating around. By day 14, they form tumoroid structures, and we start seeing stromal outgrowths with all of the cells that I mentioned before, osteoclasts, osteoblasts, and adipocytes present in the culture. And the cultures proliferate and mimic the proliferation capacity of various cell types. So here's the overall proliferation marked by loss of CFSE over time. CFSE is lost with each cell division. And this is what we're seeing, that by day 5, there's statistically significant proliferation within the culture. B cells, as known, proliferate quite rapidly, so the significant proliferation is actually seen by day 3. Plasma cells proliferate slowly, so proliferation significance is noted at day 8. And then we also see clonal expansion of malignant myeloma plasma cells. And by day 8, again, there is significant expansion, and this expansion can be dependent on the patient, anywhere 2 to 12 times. With primary lung samples that we worked with, again, if we take this tissue explant and place it on plastic, as shown right here, looking then at live dead stain with live being green and dead cells being red, most of these cells do not survive and disintegrate after about 4 days, and by 7 days, most of them are dead. But looking on the bottom here at day 27, the explant placed in our lung is very viable and actually having stromal cells growing out of it. And then looking at proliferation, looking at Ki67 staining, the ex vivo sample, so prior to culture, there is a number of proliferating cells, and at 7 days in culture, there's still proliferating cells within these explants. And then on the right-hand side, we're looking at dissociated tumor cells. Again, by day 28, they form these spheroids and stromal cells kind of connecting these spheroids, and the entire system, all of these cells are very much alive. We also looked at the presence of immune cells, and CD3 and CD33 cells are also, positive cells are also present in these cultures. So then what can we do with these systems? We can treat them with various compounds, as I mentioned earlier, small molecules, ADCs, bi-trispecifics, CAR-Ts, anything you want. And then we can look at both efficacy and toxicity in a single assay, just looking at malignant and non-malignant cells, can get rudimentary toxicity analysis, and can, of course, understand how the compounds affect the tumor cells. So one of the examples is really looking and identifying responders versus non-responders. Here we had 10 treatment naive and 10 patients refractory to pomalidomide. And as expected, The treatment-naive patients, the blue line, responded quite well to pomalidomide. These cells did not survive after pomalidomide treatment, while refractory cells from refractory patients were not really affected by treatment. Can also look at pipeline prioritization and patient stratification. So here we tested a number of compounds and combinations, looking at, say, 2 different patients. The treatment-naive patient can see dose specific response to treatment to various compounds, and combinations worked even better. But this patient B, who was pomalidomide resistant, you can see only really compound 1 in this case had any kind of effect, and even then it was significantly worse than for the treatment-naive patients. So here you can really start getting understanding of how different patients will respond. Looking at really efficacy and toxicity together, here we looked at multiple myeloma, and we looked at plasma cells where the treatment with bortezomib or another investigational compound showed a dose effect, while looking at non-tumor cells, there was really no effect of the compound on that. And finally, we're looking at CAR T cells and how can we use CAR T cells in these systems. And because the Arbone really incorporates everything in a single assay, this type of system can really be readily used to evaluate CAR Ts without really requiring any exogenous activation of these cells. So when we set up the culture and then 3 days later add CAR T cells, about 80% infiltrate into the matrix within 4 days. Antigen-specific CAR T cell activation is noted within about 2 days after cells infiltrate. And then we see also CAR T cell proliferation over the course of the culture. So really this model is suitable for looking at CAR T cell dynamics and CAR T mediated cell kill. And really what we're looking at here on the right-hand side, the spheroids, these cultures treated with mock CAR Ts versus target-specific CAR Ts. And you can see the target-specific CAR Ts induce cell death as these spheroids starting to turn red. And looking on the bottom, you can actually see CAR T cells kind of sitting on top of the tumors. And here it's these tumoroids. This is on the right-hand side is even clearer, where these cells are really interacting with the tumoroids and can start inducing cell death. And the table just summarizes what I just said. The target-specific CAR T cells, we see about activation about 17% of these cells, while the controls don't really see any. And tumor cell death was noted about 73% tumor cell death, while controls were in around 20% or so background death. So this really captures the dynamics of CAR T cell mediated kill. So just in summary, we really can look at response to treatment in tumor-specific cultures because these really correlates with clinical response and gives correct readout for really how the drug will behave. Our models can maintain long-term survival of tumoroids and stromal cells, and it can be used for evaluation of various compounds. It's compatible, as we talked about, multiple different types of compounds. Suitable for evaluation of on-target and off-target effects of various agents, and can be used to stratify patients into responders and non-responders, and of course can be utilized with all the standard readout methods. And we have created a number of different types of models. We can use immortalized cell lines, patient-derived cell lines, primary dissociated tumor cells. We can use syngeneic cells, so really various, and we have metastatic models as well. So very widely applicable technology. And we talked about multiple applications such as efficacy testing, immuno-oncology, looking at patient stratification and such. So with that, I'm going to hand it over to Dr. Ersk, who is going to talk about more how this can be incorporated into overall drug development paradigm.
I'm sure you will all agree that was a great presentation. Our next speaker is Sumitra Urs, Scientist in Scientific Development at Covance by LabCorp. Sumitra has over 15 years of experience in research and development, focusing on different aspects of cancer biology. Over the past 4 years at Covance by LabCorp, she has established new preclinical syngeneic, xenograft, and PDX models, deciphering their efficacy and unique treatment responses to immuno-oncology agents, standard of care agents, radiation, and combination treatments, to enable strategic use of the data by biotech and pharma companies, utilizing Covance by LabCorp's in vitro and in vivo preclinical services. Prior to this, she was at the University of Michigan Translational Oncology Program, managing the xenograft core. So without waiting any longer, here is Sumitra's presentation.
Hello, everyone. Thank you, Julia, for the wonderful presentation on ZPredictor's 3D platform and for highlighting the advantages of the 3D system in general, and in particular, the organ-specific ZPredictor platform. My name is Sumitra Urs, and I'm a scientist at the Preclinical Oncology Early Phase Development Solutions Covance, located in Ann Arbor, Michigan. In the preclinical setting, there is always an urgency to develop models that are predictive and relevant at the same time. Early drug development continues to put extensive efforts to develop strategies to recognize multiple avenues. That includes response to treatment, combination therapies, and to identify novel biomarkers and targets for screening large number of test agents. Not only that, since time is critical, a rapid turnaround time is an equally important parameter. So we asked ourselves, can we and how can we employ the 3D platform to Covance preclinical oncology and drug discovery program? Given the ever-increasing relevance of in vitro 3D models in the PCO space, Covance entered into a preferred partnership with ZPredictor. The partnership is to develop new specific extracellular matrix for preclinical oncology screening. As Julia mentioned earlier, the ZPredictor 3D platform technology will provide organ-specific extracellular matrix that can support tumor cell growth with the composition and architecture that is required for both solid and heme models. This positions the platform as a tool for in vitro screening of tumor models with in vivo characteristics. With this in mind, we validated the in vitro ZPredictor 3D platform at our PCO site in Ann Arbor using both human and murine models. Today, in this presentation, I will be sharing some data on the same. The methodology we adopted to validate the 3D reconstructed mouse breast specific matrix, which I will refer to as RM breast in the rest of the presentation, is as follows. We first compared growth kinetics and morphology in 4 syngeneic mouse breast carcinoma cell lines, namely 4T1, EMT6, E0771, and MMTVPYMT. Cells were cultured on regular 96-well tissue culture plates to represent 3D cultures and on 3D matrix in similar 96-well plates. Analysis for 2D and 3D was done for tumor growth, optical cell density, viability, tumor morphology, and response to treatment, and was based on cell titer glow luminescent cell viability assay readouts for both 2D and 3D. Luminescence was quantified using the Citation 3 imaging plate reader. Analysis was based on a 4-parameter non-linear curve-fitting analysis normalized to the vehicle-treated wells. Moving on to the results, this slide shows bright field images of the 4 murine breast cancer lines on the 3D matrix. The top row shows tumor cells after 5 days in culture, and the bottom row shows tumor morphology after 12 days. As you can see, each cell line has its own unique morphology and growth characteristics. The tumor cells can form clusters that are loosely connected, as in the case of EMT6 and E0771 in the middle, or form compact spheroids as seen in 41 and PYMT. Tumors form spheroids and migrate in the Z-plane and appear as free floaters. These cell lines can be maintained in culture for 15 to 20 days with minimum intervention, which offers more than 10 days window for evaluation. On this slide, I want to show specifically the comparison of 41 cells grown in 2D and 3D. On the left is a representative image of 41 cells on a regular 2D plate showing cells growing in a typical monolayer fashion. When plated on normal tissue culture treated polystyrene plates, 41 cells maintain a flattened, polarized, and proliferate rapidly to reach confluence and start to die. In the center is the image of the 41 cells grown on 3D RM breast matrix, and we can see clusters of cells branching off and forming spheroids. The image on the right is a high-power magnification of the cells in 3D. After 4 days in culture in the RM breast system, spheroids are formed from the cell monolayer and migrate in the Z-plane of the matrix. We then tested the tumor lines for their response to chemotherapy agent cisplatin. Cells were grown in 2D and 3D platform and treated with a dose range of cisplatin. Cell viability was determined using cell titer glow viability assay and the IC50 extrapolated. The blue line indicates the nonlinear curve for 2D and the red line represents the cells cultured in 3D. What we see is consistent increase in IC50 values in all the 3 models cultured on 3D platform. Cells cultured on 3D matrix have a higher IC50 value and show more resistance to cisplatin treatment than the 2D cultures. So far, we were using the cell lines for validating the 3D platform. Using the cell lines alone does not mimic the tumors in vivo, as the tumor microenvironment is still missing. So the obvious next step was to evaluate tumor lines in condition that is similar to the tumor microenvironment, with not just the extracellular matrices, but also other cell types associated in the organ-specific tumor microenvironment. We tested tumor suspension, or BRI, which was generated by dissociating fresh tumors, in this case 41, in BALC mice. Mammary fat pad tumors were excised when they reached a tumor volume of approximately 500 mm3, dissociated into a suspension, filtered and cryostored. This suspension contains all the components of an orthotopic tumor microenvironment with all cell types, including the immune cells, fat cells, stromal components, and cells of vascular lineage. The cell suspension was cultured on the 3D matrix, and after 7 to 15 days in culture, cells migrated and coalesced to form large spheroids as seen in the center and right bright field images. These spheroids were much larger in size than those formed from tumor cells alone, as seen in the previous slide. Morphologically, these spheroids are a more accurate representation of tumors and can be actually considered as microtumors. Since we were able to recapitulate to quite an extent the morphology and architecture of the in vivo tumors, we tested the response to treatment in these spheroids. We tested standard of care agent paclitaxel, shown in the left graph, and cisplatin, shown on the right graph. The black line represents the nonlinear curve for 41 cells cultured on 2D plates. The red line represents the 41 cells cultured on 3D matrix, and the fluorescent green line represents the 41 cell suspension or bright drawn-in 3D matrix platform. As you can see, paclitaxel treatment showed little to none cytotoxic effect in the 3D matrix for both cells and cell suspension cultures at the concentrations tested, indicating a degree of resistance to paclitaxel. On the other hand, cisplatin treatment resulted in cell death in all the 3 platforms tested. The IC50s are shown below in the table. Cisplatin has clear dose-dependent efficacy in both the 2D and 3D models, with the IC50 values right-shifted 5 to 7-fold in the R-MBreast platform. After seeing the difference in response to paclitaxel in the 3 in vitro platforms, we compared it to response in vivo. 6 to 7-week-old female BABC mice were implanted with 41 cells in the mammary fat pad, and tumor growth was monitored by caliper measurements. In the graph on the right-hand side, the tumor volume on the y-axis is graphed against days post-implant on the x-axis. Paclitaxel was dosed intravenously once a week for 3 weeks at 10 milligrams per kg dosage starting from day 7 post-tumor implant, when tumors were approximately 100 millimeter cube. In this slide, we show the comparison of the response of the 41 cells to paclitaxel treatment in 2D, 3D, and in vivo. Tumor cells or tumor cell suspension grown in 3D matrix are refractory to paclitaxel treatment. Under in vivo conditions, the 41 tumors grown in the organ of origin show similar response to paclitaxel and are refractory. This shows that the 3D matrix mimics the in vivo setting and shows similar response as seen in vivo in 41 model. Encouraged by these results, we are testing other chemo agents to evaluate if a similar trend is seen between 2D, 3D, and in vivo models. We have now shown that the 3D models, especially tumor suspension, mimic in vivo solid tumor biology and can show more resistance to chemotherapy than 2D monolayer cell cultures. We also know from literature that these 3D spheroids can develop hypoxic cores and exhibit diffusion profiles similar to tumors. Having validated the 3D platform for testing standard of care agents, we wanted to evaluate its potential in the CAR-T space. Testing CAR-T therapy on 3D tumors can help with screening and choosing the proper antigen targets and proceed into preclinical studies, especially as it can be fast-paced and cost-effective. We use the Z-Predictor human R-bone matrix to evaluate effects of anti-CD19 CAR-Ts against the NALM-6 luciferase-tagged human leukemia model. As a CD19 positive cell line, NALM-6 cells are ideal for testing novel CAR-T cells or other immunotherapies for efficacy. This is a graphical representation of the design for in vitro CAR-T testing using the human R-bone 3D platform. Tumor cells were plated in 96 well plates with the R-bone matrix and cultured for 3 days, after which anti-CD19 CAR-Ts generated in-house at Covance PCO were added to the cells at different concentrations and incubated further. Cells were harvested and processed after 2 days and 4 days post-CAR-T treatment to determine live T cell counts and analyze for live dead target cell staining by flow cytometry analysis. The top panel shows flow cytometry analysis after 2 days and the bottom panel for 4 days post-CAR-T treatment respectively. On the left is the live CD3 positive T cell counts for untransfused and anti-CD19 CAR-Ts at different ratios. On the right is the graph showing percent cytotoxicity, in this case the CD19 positive NALM-6 cells. Target cell killing was detectable as early as 2 days. Day 2 and Day 4 show consistent NAM6 tumor cell cytotoxicity at CAR-T ratios of 3:1 and 5:1. So, to summarize, Covance has successfully validated in vitro the reconstructed tissue-specific platforms from Z-Predictor, the RM breast and the human R bone. And in our hands, cell and tumor suspensions can be grown and maintained longer than 2D cultures, allowing at least 10 days window for efficacy evaluation. We also show that the platform can be used for screening small molecules, antibodies, and CAR-Ts. The Z-Predictor 3D platform can be used for multiple types of assays such as flow cytometry, cytokine analysis, and the spheroids can be harvested for immunohistochemistry evaluation. Put together, these 3D specific matrices can be potentially used to predict in vivo responses and used as a tool to bridge the gap between lab and in vivo preclinical studies. This loops us back to the question we asked when we started this part of the presentation as to where does 3D screening fit into the preclinical oncology space? To that end, it'll be helpful to understand the functioning of the PCO. The Covance PCO is built on technology and is supported by what I would like to call the 5 pillars which form our toolbox. Starting on the left side are the tumor models and animal strains. We have over 300 human and more than 50 mouse tumor cell line models spanning more than 30 different cancer histotypes. This includes over 100 luciferase-enabled cell lines as well, which help support use of our metastatic, disseminated, and orthotopic models. From an in vitro perspective, we have a full suite of relevant assays. The most commonly used is our industry-leading flow cytometry. We have two 14-color capable and two 21-color capable instruments that we use primarily for immunophenotyping and immuno-oncology cell-based functional assays. The Z-Predictor 3D technology very aptly fits into the in vitro services technology pillar. Outside of flow, we also run lots of other cell-based assays such as proliferation, ELISpot or fluorospot, ELISAs, Luminix, MSD, and IHC services. Our in vivo services are run in ALAC accredited vivariums by professionally experienced staff having a broad preclinical oncology experience, including surgical expertise for a wide range of orthotopic models and routes of drug administration. These skills allow the team to run a wide array of study types that are relevant within the preclinical oncology space, including our capabilities for ABSL2 level studies. One area that has been growing rapidly for us is work with CAR-T or other adoptive cell therapies. To date, we've run more than 400 CAR-Ts in vivo studies since our first one at the end of 2015. Finally, to round out our services, small animal imaging capabilities that include BLI, fluorescent imaging, and PET have been a core service at the Ann Arbor site. Therefore, as you can see, we have a large array of tools to provide preclinical oncology support throughout the drug development process. Finally, I want to thank all the people behind this work. Mike Steffi, Andrew Karlovitz, Natalie Zerba, and Scott Wise, our executive director at the PCO, have been instrumental in everything that I have just presented. And of course, I have to thank Julia Kirsner and Inkyu from Z-Predictor for all the technology and the know-how. Thank you.
I'd like to thank Julia and Sumitra for the presentations just now. We will now be moving on to the Q&A. So if you would like to submit a question, please do so in the question box and please indicate who your question is directed to. I would now like to invite Julia and Sumitra to the stage so we can begin with the Q&A. Please both join me on stage now. Hi, Paul. Thank you for joining me for the Q&A. Before we start, I'd like to remind everyone that they can submit their questions using the questions tab at the bottom of the webinar dashboard. So I'd like to start with a question to Julia, but Sumitra, please feel free to jump in and give your point of view too, and same for you, Julia, when I ask questions to Sumitra. So Julia, this question was asked by someone in the audience. How can you differentiate the tumor cells and the non-tumor cells? Can you use a cell-traced dye?
Well, thank you very much for the question. So definitely, yes, you can use cell tracer dyes. We have done various colored cell tracer dyes that can then be run on a flow cytometer. One caveat with those, you have to use the long-term dyes because these cultures are usually 7 to 14 days, depending, of course, on your compounds. And these dyes, some of them tend to get diluted when cells divide. So that really depends on the dye. Other ways to differentiate the cells is, of course, if we're talking about a co-culture of, say, tumor epithelium with stromal cells, you can just look at actual markers and differentiate epithelium from stroma or from immune components that way. This can be done by flow cytometry, can be done by imaging. So there are multiple ways of differentiating what cells are actually growing out in the culture.
Thank you, Julia. So I see we have some questions coming here. So Sumitra, do you expect any issues with oxygen diffusion with your 3D matrices?
You mean in the spheroids? Because, I mean, as such, we have not done any tests to evaluate that, but since the size of these spheroids are about 100 microns, I do not expect to see any, gradient as such. Maybe when they get bigger, we might see them, and when we keep them for a longer period of time. And
if I can add to this as well, I completely agree with what Dr. Ur said. Usually when we've looked at spheroids kind of as they're distributed within the culture, and we don't see, and as Dr. Ur said, we did not measure specifically oxygen saturation, but we don't see, for example, more spheroids dying towards the bottom than towards the top. So that kind of distribution is fairly uniform throughout the culture.
Thank you. And Julia, does this tumor-specific spheroid co-culture with MSCs also have vascular endothelial cells?
Currently, we have not added endothelial cells, but we have done assays separately with endothelial cells. So this is definitely something that can be done and added together.
Thank you. Sumitra, we have another question for you. Have you used 3D tumor cells implanted in mouse and observed the growth pattern and image or treated with PACLI?
Can you repeat it? Because I was asking if we implanted the spheroids into the mouse.
Yeah, have you used 3D tumor cells implanted in mouse? Yeah.
No, we haven't yet done that, but that's a good way to proceed, just to make sure that these spheroids or tumoroids are actually viable in vivo, and we can also look at their growth kinetics once we put them in, and then correlate them to regular cell implants that we usually do. So we haven't had the necessity so far, but if somebody is interested, we definitely can do that.
And
so Oh, I'm sorry, I was going to add something to this. Yeah, this hasn't yet been done with us together with Covance, but our group has done these studies where we looked at both breast cancer and multiple myeloma, and we would get the cells from the matrix so they can be viably isolated from the ECM and then implanted into the animals. We have done a number of metastasis-specific studies in breast cancer looking at metastatic cell population that we were able to isolate from our culture versus the kind of this tumoroid primary tumor-like population, and then put those into animals, and we would see growth patterns that correspond to the stage. So if those cells had metastatic properties in culture, they were metastatic in mice. If they didn't, then they behaved more like primary tumors in mice. And we have done implantations also post-culture of multiple myeloma straight into tibias.
And how do you prepare single cell suspensions from 3D culture for flow? I think this one is addressed to Sumitra.
How do we prefer 3D? Say it again.
How do you prepare single cell suspensions from 3D culture for flow?
We do enzymatic digestion, just like how you would do for regular tumor dissociation using the kits.
So, and also to add, for the bone marrow system specifically, we actually have a way to do the non-enzymatic digestions where you can actually isolate the cells without any kind of enzymes. For systems with epithelial cells, as Sumitra said, it's for us, we do a combination of kind of our non-enzymatic digestion, and then some of the spheroids are fairly tight, so those sometimes do require a bit more of an enzymatic treatment, taking care not to, of course, lose any surface antigens, but most of our digestions are done just to depolymerize the matrix.
Thank you. So now I have questions which I'm not sure who they are addressed to, so please feel free to jump in once I ask them. So you are using collagen or matrigel for the formation of the original cultures. Do you expect that to alter cellular response to drugs, since these are not natural part of the ECM?
Can you please repeat the beginning of the question? I didn't catch the first part.
Yes, you are using collagen or matrigel for the formation of the original cultures. Do you expect that to alter cellular response to the drugs, since these are not natural part of the ECM?
So all of our matrices are formulated to incorporate the components of the extracellular matrix that mimics the particular tissue. If collagen 1 is not part of the tissue, it will not be in the system. If collagen 4 is not part of that tissue, it will not be in the system. So it is formulated specifically to match the tissue of interest. Therefore, what we see, the drug behavior that we're seeing matches what we see in the patient, as I showed kind of early on in my presentation with some of the clinical studies that we've done.
And have you ever tried treating 3D platform with ASO, short oligos, or exosomes? What is your experience with them? Any hurdles?
We haven't done exosomes or oligos on their own. We've done a lot of different molecules. These are just haven't been, we haven't had any experience yet with them, but we have done different size molecules, different types of molecules. So I don't foresee this not being that we wouldn't be able to do it, but it's something that would have to be tested.
In tumor explants, how did TILs grow during culture time? Did lymphocytes propagate proportionally?
So one of the experiments that we've done with the lung tumor cultures that I showed, where we did look at some of the immune cells that were kind of came with the tumor, there was proliferation of infiltrating cells, but we haven't yet assessed, this is an ongoing set of studies, we have not yet assessed the kind of proliferation compared to epithelium or compared to other compartments.
And Sumitra, this question is for you. What downstream analysis can be performed with the spheroids?
We can do multiple. For one, we can do flow, like we already discussed, and then we can always harvest them, embed them, and then do histology. Use it for any analysis of cell type, distribution, and basically look at other markers if we want to. So that is an advantage too. It's as good as using a tumor with just a smaller tumor.
And do you plan to extend the models to rats and hamsters?
That's a good question. At the preclinical oncology in Ann Arbor, we use mostly mice and rats, but not hamsters. But if that is something that someone's interested, we could definitely look into developing it with using the larger Covance capabilities.
Thank you. And Julia, there's another question for you. Can the treatment be an oncolytic virus?
We haven't tested it, but yeah, it can be. We have actually done for a completely different purpose. We've done some studies with viruses in general, and we get infection of the cells in these systems. So yes, it's possible to do that.
And we have another question coming through. I'm not sure if it's for you, Julia, or for you, Sumitra, but how did you determine the ECM of the in vivo original tumor?
So this is something that really is based on our kind of central technology. So we evaluate the tumor composition and base our formulations on that. The exact procedures are unfortunately not something I can share.
And will your organospecific ECM be commercially available?
So it is available from both Covance and us as a service. We right now don't sell the product, but this is definitely something that we can talk about. So definitely feel free to reach out to us.
And another question we have here is, have you ever tried to use patient-derived tumor extracellular matrix, maybe with the help of an hydrogel?
So because our technology is specific to individual tissues, this is the centerpiece of what we're doing. So we do not like including hydrogels or any kind of synthetic materials in our system. So we're using our own formulations.
And Sumitra, would you recommend the use of tumor suspensions over cell line cultures?
Definitely, at least in the syngeneic models that we have worked with, they definitely showed a better response that correlate more to the in vivo responses that we see. So I would say yes. That was my take. What is your opinion, Julia?
Yes, absolutely. Anything that has native microenvironment that can be brought into the system is of a great advantage, because our matrices provide support for these cells to grow and to survive and to maintain the heterogeneity of the original tissue. So bringing in the original tissue in its entirety is much more useful than just having, say, a cell line on its own. And definitely an advantage of this system.
Sorry, in a way, it also has its own, structuring when we dissociate it into BRI, which is not actually single cells. So, there are small clusters, and then you have the advantage of having, cells from which are vascular lineage. In syngeneic models, you also have the immune cells in them. So it definitely would be an S for that answer, for that question. Answer is yes.
And we're running out of time, so I will read now the last questions, and all unaddressed questions will be passed on to Covance by LabCorp. So have you compared your results with Matrigel?
Yes, so we've been doing benchmarking studies and comparing the Matrigel to our formulations. Usually for cell lines alone, it depends on a tissue type. So if we're looking at tissues where components of Matrigel are really not present, like if there's no collagen 1 or laminin, which are the main components of Matrigel, things look very, very different, of course. for simple cultures with cell lines alone, if you, for example, look at breast cancer, where the basement membrane of mammary tumors is mainly collagen 4 and laminin, there is, of course, not going to be a huge amount of difference. Where you start seeing a lot of difference and true representation of the tissues when you also start adding stroma to the system, and cells within our formulations really proliferate and expand and survive based on their native capacity in the tissue, which is tends to be skewed in nature gel.
Thank you both. As I said, we're running out of time, and all the questions that haven't been answered will be passed on to Covance by LabCorp so they can address them directly. Thank you to the audience for submitting so many questions, and thank you both for your answers and earlier presentations. Please feel free now to leave the stage. That is sadly all the time we have, but I hope you enjoyed the session. As I said, all unaddressed questions from today will be passed on to Covance by LabCorp. Please look out for an email from us tomorrow, as we'd love to hear some of your feedback on today's webinar. If you do have any follow-up questions or comments, please email this to myself at info@Hanson-Wade, and I will get back to you soon. On behalf of Covance by LabCorp and CDX Engage, thank you all for your time, and I hope you enjoy the rest of your day. Hope to see you next time.